just advance it up on the
Uh-huh.
computer,
Okay.
whatever you're comfortable with.
Okay. Wow. Sounds fantastic.
It was a gift. And then I had to rebuy it because I left it at some place I went to give a seminar. Laser pointer, fancy. Cool. I took the Princeton one. Me too.
That's
I
pretty
know.
good.
It's up for debate, but
Hi, everyone. My name is Heather Bean. I'm an assistant professor in the School of Life Sciences at ASU, and I help to organize the Life Sciences Cafe. I also want to extend a welcome to our online audience that we have today. So you're going to see that we're going to be all mic'd up when we do Q&A later on. I'll be repeating every question that you ask so that our at-home audience can hear the question. But I appreciate you coming here and being part of the audience because hopefully we're going to have a really vibrant discussion today. I want to Next week's seminar, we'll be having Fei-Wei Li joining us, who studies the coevolution of ferns and cyanobacteria, and he'll be talking to us in Life Sciences Cafe next week. Today, what we have for you is a panel discussion on personalized medicine. This is part of our goal to bring hot topics in science to our school and also engage a broader community at ASU. So this is a collaborative discussion between the School of Life Sciences and the College of Health Solutions at ASU. And today we have Stefan Johnston with the School of Life Sciences, and we have Beate Peter and Cori Wisner, who are both with the College of Health Solutions, join in our panel discussion today. So what I want to do is start you off with a little primer on what personalized medicine is, just to lay the framework for with some definitions, and then we're going to have 3 short talks by our panelists so that they can tell you about what they're doing in their research when it comes to personalized or precision medicine or diagnostics. And then we're going to have our Q&A with all of you. So let me start you off with a little bit of a definition, just give you a bit of perspective about how we typically approach medicine when it comes to, for instance, the approval of a new drug. These drugs developed by pharmaceutical companies, when they go to clinical trial, they collect a group of people who they hope to be diverse and representative of a disease, and they will give that medication to that group of people and measure the outcomes. And typically what they'll see is something like this, where maybe 25% of the people that they dosed with that medication, in this example, were not responsive at all to that medication. That medication did nothing to help them with their disease. And then maybe 75% of the people who received the medication at this particular dose did have a response that was favorable. That medication did help, but it helped to varying degrees for every single person that did respond to the medication. And the real challenge with this approach is that we can't predict out of this group who's going to be responders and how well they're going to respond and who's not a responder. That next patient comes and they're going to be treated with this medication. We can't predict the outcome of that therapy. And that leads to a lot of wasteful time, basically, in helping people treat their diseases. It also can lead to a lot of unintended side effects and consequences. If that patient's not going to be a responder, they aren't necessarily also going to be free of any side effects that that medication can impart. The precision approach is that with some combination of information about that person's genetics, about that person's lifestyle, maybe their exposures from their environment or from their work, you can utilize the information to predict what treatment approach for that individual patient would be, what sort of population they would fall into. And in this example here, maybe Mr. Orange will get the best therapeutic response from the standard dosage of the medication. And that could be predicted by this combination of information. But Miss Green and Mr. Purple need different doses to see the most beneficial therapeutic effect. And Miss Blue needs an entirely different treatment approach altogether. Miss Blue would fall into the non-responder category under the standard therapy. Again, the precision approach is that the idea that we can utilize information about these patients to help predict the response that they're going to have to a particular therapy. And we can identify the best therapy for them utilizing that information. Now, the title of this seminar was Personalized Medicine, and what I was just describing for you was precision medicine. So are these terms the same? Is it potato, potato? They are used interchangeably a lot, and it will probably depending on who you ask. But in 2011, the National Research Council did a study to figure out what people understood about precision medicine and what they understood about personalized medicine. And the responders of this survey stated that when they heard the term precision medicine, what they understood it to be was what I just described to you, that there could be some information about that patient that could be utilized to help predict what that patient's best option was going to be for therapy. But when it came to the term personalized medicine, what patients thought of was this, that a patient, or what survey responders thought of was this, that a patient would go into the doctor, that some information or maybe a blood sample would be taken, that'd be sent off to a think tank, to a group of scientists who were going to be able to do some work based on that patient's samples and create a drug just for that patient. And in 2011, this was not a standardly available approach. And so it was decided by the National Research Council at that time that we really should use the term precision medicine when this is the approach that we're talking about, that we utilize data to predict a patient's best response. But personalized medicine is a thing and is being used to good effect, although it's quite expensive to do so, for some diseases such as cancer therapies, where we have chimeric antigen receptor T-cell therapy for lymphoma, where a patient's blood sample can be taken and sent to a team of scientists who can then extract the T-cells. They can engineer that patient's T-cells to fight that patient's cancer. And then those T-cells can be administered back to the patient to fight and kill the cancer. So this is truly a personalized medicine approach, and this approach would not work for other patients. It's individualized to the patient. When it comes to precision medicine, there is a lot of enthusiasm about this, which is why we chose it for Hot Topics for today. As part of evidence for this, the National Institutes of Health are all in. They have developed this program that's called All of Us. It's a huge precision medicine initiative. They are enrolling 1 million subjects to collect specimens from and to collect information. They're collecting blood, urine, saliva, I think feces, all sorts of biosamples. They're also collecting information about that patient's history, what diseases they have, what their disease families. They're getting genetics information from that patient. And they're putting this into a huge database so that they can start to identify some approaches about how to prevent disease on a personalized level, and also about how to treat diseases and diagnose them as well. So this is a huge effort that our government is putting a lot of money into to drive precision medicine forward. At ASU, we are also heavily invested in precision medicine, and we have a lot of efforts going on campus And I've had invited 3 panelists to join us for a discussion today to tell you about just their efforts on campus on precision medicine or personalized diagnostics. And today we have Beate Peter, who's an assistant professor in the Department of Speech and Hearing Science, and she's on the Tempe campus. We have Cori Wisner, who's an assistant professor in the School of Nutrition and Health Promotion on the downtown campus. And we have Stephan Johnston, who is the director of the Biodesign Center for Innovations in Medicine and a professor in the School of Life Sciences. They're now going to each introduce us a little bit to their research and how it applies to precision or personalized medicine. So I'm going to welcome Beate, please. The mic is on.
All right. So if you can't hear me, it's probably because of the German accent. Okay. Okay, I'll move here. All right, there's the title slide. So I decorated the title slide just a little bit with some nucleotides here. I'd like to draw your attention to this little sequence here because it'll come up in just a couple minutes. There it is, TGCA. I had the great privilege of being at the American Society of Human Genetics meeting last week in Orlando, and I was in the audience when Bill Gates and Francis Collins talked about precision medicine. It was fascinating. It was the most interesting talk, 90 minutes. I was just riveted. And so their themes in precision medicine are truly medical themes. They talk about HIV drugs and vaccines. They talk about cancer, flu vaccines, non-communicable diseases, these kinds of maternal health. Bill Gates is an extremely impressive individual, I have to say, with his passion and his knowledge about these global public health issues. Collins said that precision medicine does not imply that you have to develop one drug for one person, but rather, if you have a menu of interventions, you try to figure out which one is going to work best for which of the many thousands of people who need that particular intervention. This is exactly what Heather had just outlined for you. And Gates talked about genetic interventions in the future, potentially for sickle cell disease. So these are 2 thought seeds that I wanted to pick out for you. In my lab, I do something completely different. I'm in the Department of Speech and Hearing Science, and I study the genetic etiology of speech disorders, language disorders, dyslexia, stuttering, anything to do with communication disorders that falls into my realm of interest. Why is it important? Why are these kinds of disorders important to me? I just have a few quotes for you from parents with children who have these kinds of disorders. And they are rather desperate because these children are many times of completely typical cognitive ability, but they are unable to share their thought life because people can't understand them because their speech system isn't online yet. That is very, very frustrating. It has a devastating effect on a child's emotional and social and educational development. One person who enrolled her entire family in my study said it best. She said, "Thank you for doing the work that you do, so in the future, perhaps, other children with these kinds of disorders can receive help earlier than our child." And so this woman, in a way, she formulated precision medicine for me as applied to the behavioral and clinical disorders that we study in our college and our department. So what do I do in my lab? I take very extensive behavioral data and not just standardized tests of speech and reading and fluency or these kinds of things, but also innovative ideas, because I've noticed that, for instance, some individuals with apraxia speech and individuals with dyslexia not only struggle with the speaking and with the reading piece, but also some fundamental underlying deficits. For instance, handling sequential information is very, very difficult for them. That's an underlying current, not just on the surface of the phenotype. I have an EEG system in my lab that I use, and with colleagues, I get MRI scans to look at, for instance, cerebellum. And we send off our DNA for genomic analysis. For instance, in multi-generational families, we look at how are these disorders inherited in the families. In trios with sporadically affected individuals, we look, what does the offspring have that neither mom nor dad have? Or could it be compound heterozygosity? Sorry, stuttering myself. And so the big idea is, having a genotype-phenotype association that takes into account the broad set of phenotypic behaviors that we have, as well as the genomic variants that we find. And in the future, there should be a clinical translation of all of this. So can we define subtypes of these disorders on biological grounds rather than by some test or some behavioral metric? Can we develop subtype-specific interventions, very much so like the warfarin effect, where you have this genotype, we're going to give you this dosage, kind of. So can we look at somebody's biologically relevant phenotype and give them a specific therapy? Early identification, very, very important. Dyslexia is not diagnosed until 2nd or 3rd grade. By then, years of difficulty with sequential processing have gone on, but unnoticed and untherapized. And so early identification and developing preventative strategies, that's my dream. And maybe someday, like Francis Collins and Bill Gates were discussing drug treatments, maybe gene therapy. That's in the way, way, way distant future in my mind. And so these kinds of things have led to recent publications. So from dream to action, the Babel Bootcamp is something that we started earlier this year. We focus on infants with galactosemia because in their case, it's already known that if you have galactosemia, most likely you'll end up in the speech pathologist's office for language therapy and speech therapy. And that's just a little piece. And so we thought, typically they don't get any interventions unless they show a deficit already. Let's work with them while they're even still learning to babble, because many of them never babble. And so our question is, if we help them babble, if we help them develop pre-speech vocalization, will that kind of grind the engine into action for later on? So this is one of the things that we do in our lab. And here comes a few plugs. We have in our department now a new PhD concentration called Translational Genetics of Communication Abilities. There's the TGCA that I alerted you to earlier. And behavior genomics at ASU, I was recently awarded funding for a Team LA group to investigate whether behavior genomics at ASU can be driven forward and implemented in some way, shape, or form. I'm working closely with the educational leaders at the American Society of Human Genetics. Michael Dougherty is my colleague there, and Kate Reed is my colleague at the Jackson Laboratory. And sorry about the typo, didn't catch until today. And so what we're doing next month, in just a couple of weeks, we're doing a 2-hour seminar on genetics for clinicians. So spotting red flags for potential genetic etiology and making the right referrals. So connecting the world of speech-language pathology with the world of genome professionals. And that is my 5-minute talk.
And next up, we have Cori Wisner, who's going to tell us about her research.
I bring bad omens when there's technology. Okay, thank you. All right, well, thank you everyone for being here, and thank you for the opportunity to speak, Heather. So I come from a different area of the College of Health Solutions. So my background is all in nutritional sciences, and to be honest and in full disclosure, I'm not an expert at all in personalized or precision medicine. I have interests that maybe push into the realm of that with regard to nutrition and how food and functional nutrients and functional foods may play a role in optimizing growth and development in early life. And so that's sort of where my research comes from, and specifically one of the things that we have going on inside of us in terms of genetics is not only our own genetics, but also the genetics of the writhing mass of bacteria that live inside of our gut, who also help us optimize the nutritional components that we eat so that we can absorb them better and grow and develop better. So one of the areas that I have the most experience working within this realm in terms of the gut microbiota and nutrition is with regard to bone health and mineral metabolism. So throughout life, we are constantly turning over our skeleton. About every 3 years, you actually reproduce your entire skeleton because it's always changing. It's quite a dynamic tissue. And most of us, when we think about osteoporosis, tend to think of individuals who are elderly and frail, but actually what happens is that there's this window of opportunity during pre-adolescence, so 12 to 14 for girls and 13 to 15 for boys, where your bones are like a sponge and you actually acquire a quarter of your adult skeleton in that 2-year period. So if you think back to that time in your own life, maybe you didn't eat as well as you possibly could have. And so we know that in the nutrition world that we can set recommendations that meet 97% of the population's nutritional needs, but the likelihood that people actually meet those intakes is slim. So what can we do to optimize absorption of nutrients when we know that dietary intake is lower than what it should be? And so this is a really great depiction of some of those things. So here we have someone who meets their calcium intake, they reach their genetic potential for peak bone mass, whereas someone who has inadequate calcium intake does not reach that peak. And you go throughout your adult life kind of maintaining that in right around age 40 to 50, especially in women where you have the postmenopausal decline in bone mass. You start to dip down and lose bone over time, but you see in the adequate diet, they're prolonging that decline longer, and where the inadequate diet occurred early in life, you have that drop into osteoporotic levels where you have a risk of fracture. So obviously, promoting more milk consumption can be challenging in the early years. So one potential mechanism for addressing that is to add something to the diet that just increases absorption of calcium that's already there. So you don't necessarily have to add more milk, especially if that's among a population that's not really interested in drinking milk. They'd rather drink soda. And there is a compound called a prebiotic. It's a type of soluble dietary fiber. They're naturally occurring in the foods listed here, which most of us don't eat a whole bunch of every day. But they're naturally occurring in things like chicory root. We can extract inulin. We can extract fructooligosaccharides for any of these products here. And we can also synthetically make them by feeding non-digestible fibers to microbes, and then they turn them into useful products like galactooaligosaccharide and lactulose. And these products, they cook into everything. They're soluble, so you can add them to liquid, you can bake them into a muffin, you can put them into a fruit snack gummy. The possibilities are endless, really, from a food science perspective. So really easy to hide in things that adolescents do eat. So my work has been working with specific types of these fibers to study how they affect calcium absorption during growth. And so in this chart here, you can see the fractional calcium absorption, the fraction of what's consumed that actually gets absorbed into the bone. And you can see that after 24 hours, we don't really see a difference between the soluble corn fiber treatment and the control treatment. But by 48 hours, we do see a significant difference. So we can optimize absorption, and the fact that it doesn't happen until 24 to 48 hours suggests that it's actually the gut microbes in the lower gut or the colon doing this work. And if you just look at the averages for this, you can actually say that this increases absorption by 12%, which is, it doesn't sound super awesome, but if you could do that over a whole year, that's about 2 to 5% of your adult skeleton that you've accrued then over a year. And we know that then the time is consistent with gut absorption, so that helps us get at the mechanism that we could optimize this in populations. And we know that we see specific changes in microbes that ferment fibers and carbohydrate material. But oftentimes this is what you would see in a paper. They just give you the mean and the standard deviation, and you go on about your day thinking, oh great, these might help something. But what happens if you plot every single person and look at their response? And this gets at what Heather mentioned in terms of responders and non-responders. So we see this for nutrition too. So here we have a group of individuals in the study that responded, and you actually see that they increased by about 20% their absorption. So that's now about 10% of their bone per year that they could earn in addition to what they were already getting. But then we have non-responders, and it's not just that they don't respond to the treatment, they actually suffered a deficit in absorption. So now we're taking a population that we know habitually consumes too little calcium, and we feed them something that we think is helpful, and that also actually is showing up in the food supply, unbeknownst to consumers, that could actually be hurting them during this incredibly important period of growth and development. And so my questions get more at what is different between the responder and the non-responder, and can we turn a responder or a non-responder into a responder? And so my last slide is actually kind of more related to the mechanism, because this is where you can then build in different ideas of trying to get a non-responder to be a responder. And so this is my little cartoon of what we currently think is the mechanism by which these fibers work. So we have soluble corn fibers. So here's our little fiber that comes in, and then we have our resident microbes that ferment it and produce something called short-chain fatty acids. These are really important because they're the primary fuel source for our intestinal cells. They have anti-inflammatory properties, and they're acid. And acid solubilizes minerals so that we absorb them more. So the more acid that's there, the more likely we are to pull calcium off of anything negatively bound, like this, to free it, and then this is the form in which we absorb calcium. And then they can passively diffuse right across the intestinal lining. And so right now we're working to analyze our data in a different way to see, are these little guys missing in the non-responders, or are they there, but maybe there's competitive inhibition where they're not able to actually ferment and carry out these fermentative processes to optimize absorption. So my personalized nutrition journey is sort of just starting, and this is where I'm using all of this data to move forward in that area. But this applies in many, many aspects in terms of nutrition and how people respond. And we all eat, therefore this affects all of us, I think. So thank you, Heather.
Good afternoon. I don't have any slides. I know we came here to praise precision medicine, but before we bury non-precision medicine, I'd like to just remind people of a few things. The most important medical impact in terms of human existence has been two things: vaccines and antibiotics. And those were very non-precision. And they are continuing to save lives. As we speak, a good share of the world is being cured of hepatitis C from a non-precision drug. And so there's huge advances being made. But I do think, and it sounds a little curmudgeon-y, but most of the history of precision medicine has been largely driven by unfounded exuberance that was not very analytical about what the possibilities and limitations were. But it doesn't mean that there isn't a lot to come out of it. And I'll try and walk through one area that is related to what I work on, and to give you an example. So as you know, we had precision medicine really started with its vision with the sequencing of the human genome. And this was one of the rationales for doing it, is that we would be able to sequence people's genomes and be able to get some insight into the diseases that they have and do something about them. That's largely turned out not to be the case. And you could have known that ahead of time, that most chronic diseases are multifactorial. There isn't a simple silver bullet that you can use for them. And we still have problems with treating chronic disease. Then the emphasis moved a little bit more towards sequencing tumors, that we would now largely focus our precision medicine efforts on treating cancer. And that led to large sequencing efforts on cancer sequencing tumors. We've now sequenced hundreds of thousands of tumors. And that initially was largely driven by the concept that we discover another Gleevec. Everybody was looking for the next Gleevec, that you'd find something that occurs in tumors where it creates a new tumor-specific target that you can drug. And Gleevec was a wonderful inspiration for that. And there's been a couple examples since then, but they're pretty rare and far between. So that didn't really pan out. So the next level of enthusiasm was the idea of precision medicine as you were portraying it, that you will sequence somebody's tumor, you will find the mutations that they have, and you will either create a drug, ideally, but at least know which of the many drugs that are out there that you would specifically give to that person. And clinical trials have been done. They're still ongoing, large clinical trials doing this kind of effort. And actually, the story is still not resolved as to whether this has net clinical benefit to do these kinds of things. There are specific examples where it's worked wonderfully. They discovered some mutation that somebody had some rare drug that was on another clinical trial, and they were able to enroll them, and they have a miraculous response. But it's been few and far between. And so it's still the story's out on that. It's expensive to do, as you can imagine. So, and what's also happened, and this is ironic, is in that time period where people were doing these precision medicine ways to create your drugs, the checkpoint inhibitor revolution took place. So all of a sudden, people started treating cancer with checkpoint inhibitors and finding even more astounding responses than you'd ever seen with drug treatment. And it was very non-precision. Everybody got it. Anybody that failed treatment got one of these checkpoint inhibitors. And now it's a multi-billion-dollar, $20 billion a year business out there. And it's in almost every clinic, in standard clinics now, they're using checkpoint inhibitors. The value, the clinical value of the checkpoint inhibitors, this non-specific approach to cancer, is hundreds of times that seen by precision medicine and drugging so far. So clearly a non-precision thing, one hands out for a precision approach. But that's not the end of the story. And this comes into something we're interested in. So those checkpoint inhibitors, by giving them to all these people, they actually started to figure out why they worked. And why they worked was because your own immune system is responding to neoantigens, mutations specifically in your tumor. And so that spawned a whole new industry now that's called personalized cancer vaccines. And this is personalized writ big, as you had in your slide. The idea is that you go in and sequence somebody's tumor, you discover their mutations that are specifically in their tumor, you actually sift through them in some way to predict which ones are going to be good antigens, and then you manufacture a vaccine specifically for that person. And after you're done, you throw it away because you'll never use it again. Because all of the neoantigens that people have found in tumors are personal. Almost every one is personal. Your tumor, you sequence it, those mutations will not exist in anybody else. So a company like Moderna now just completed a 90,000 square foot facility to build personal cancer vaccines for everybody that as they come through. And they're even moving these cancer vaccines into an earlier stage that you don't have to have failed other treatments in order to get cancer vaccines. And it is very exciting. The first clinical data is just starting to come out. It looks very promising. It looks more promising than anybody had thought before. But there is a caveat. It is very personal, but it's very expensive. And you've got to remember that 70% of the cancer that occurs in the world occurs in developing countries, not developed countries. So the projected cost for these personalized cancer vaccines, which they probably will combine with checkpoint inhibitors, are thought to be somewhere in the $200,000 per course of treatment or more. So people in Uganda who have cancer are not going to get this as a treatment for their cancer. And so I think that's often lost in the discussion, is that personalized almost always means expensive in the modern developed countries' discussion of this. And so we I think, have to start to consider personalized relative to inexpensive so that other people in the world can do it. So in that vein, I know I'm supposed to talk about what I do. I'll finish up with just one example. So the reason these personalized cancer vaccines cost so much is because they have to sequence your tumor, manufacture something from scratch for you, and that many of them are RNA vaccines that are going into people. So they have to have these special facilities, manufacture them, get them back to you. It takes 3 months to do that right now. And so they think they can get that down to 1 month. So you're sitting there with your tumor waiting for your vaccine to be manufactured. So what we said is that maybe we could revolutionize this whole process and make it cheap. So we started looking for neoantigens where nobody else had looked for them before, in the RNA, not in the DNA. And it turns out that they were there, and they're common between tumors. So now we can take a drop of blood, only we've done this in mice, not in people, but take a drop of blood, put it on an array of peptides that represent these neoantigens, look to see what you're reactive to. We've already pre-made these because it's a limited number. Give it back to the mouse, and about 50% of the mouse will be cured of their cancer. And the cost for this is minuscule relative to what people want to do for the personalized cancer vaccines now. So that, I think that's the kind of approach in personalization I think we have to have. We have to be asking, what can be personalization for everybody, not just for people that can afford $200,000 treatments? Thanks.
Okay, so now we're going to open up to questions from all of you. And we'll have all three of our panelists up here. If you guys will pass the microphone as you have something to share or your thoughts on a particular topic. And I'm going to check that the microphone is on. It's on. Okay. Okay, so we already have a question. Awesome. Go ahead.
Professor Johnson, you say currently things are very expensive, as you mentioned, but isn't it also true that in the history of medicine that as it becomes more developed, more produced, that the costs drop? So is advancing technology and volume production going to lower the cost of the
And I'm going to, so go ahead and repeat the question for our
Okay, so
online
the question,
audience.
yes. So the question was, yes, these are new treatments and they're expensive now, but as it progresses, it'll go cheaper. That's generally been the case, but for drugs, only when it's fallen off the generic cliff, right? Most of the times, the drugs are very expensive. I think an argument you can make is for something like hepatitis B, or a hepatitis C drug, which was in the news very much because I think it cost $80,000 per course of treatment. But if you actually do a cost-benefit analysis on was it worth spending that $80,000, it undoubtedly was for these people. So yes, I think it could get cheaper. The personalized cancer vaccine, that's going to be tough because of just of what the costs, the embedded costs in the manufacturing are for this. The checkpoint inhibitors, yes, I think they might come down in cost, but it's so competitive right now, I don't think that's going to happen for some time. So for us, our solution to that, I didn't mention this, but we also are working on a general preventative cancer vaccine, a vaccine to prevent anyone from getting cancer, which also works in dogs. So that, I think, is another approach to where non-precision may actually be a better approach in terms of addressing this cost than other things. Is that okay?
Yeah, please go ahead if you have different thoughts on either nutrition, develop personalized approaches, and the costs of that for nutrition or for speech therapy. Do you see that the implementation, the development phase might be more expensive than the implementation phase down the road?
Well, I think in our world, if we develop behavioral strategies that we try out on small numbers of people first and then implement them in larger, I think the development of it, the clinical trials will cost some money. I don't think that we're necessarily giving drugs to our people, but we want to find precisely targeted behavioral interventions and preventative behavioral interventions. And we find something that works for different subgroups very well, then that's what we will do. And yeah, I think initially there will be more costs involved, but later on it will be paying for itself, like in your case.
I think in terms of nutrition, things are already happening where foods are being developed and supplements are coming out. The supplement industry is a little bit on the pricey side, but I think that with so much competition, other companies getting on board drives the price back down, and there's usually options that are financially sound for pretty much anyone, but I think it's still a little bit more expensive.
Can you talk a little bit about non-responders versus responders? I know you're trying to figure out SCF's distribution, but genetic makeup, population makeup, what's the difference between responder and non-responder?
The question from Rajiv was to ask Cori to talk a little bit about the responders and non-responders in her study. What is driving those differences?
So in terms of bone health outcomes, the concept of responders and non-responders has largely been ignored, actually. There are only a few papers that have even really brought that idea to the forefront, and one in particular has tried to look at some genetic differences, and so they find differences in the FOC1 gene, which codes for a vitamin D receptor that upregulates these calcium transporters that allow calcium to move through the cell. And there are some genetic differences, but again, it's basically an N of 1 to understand how the prebiotics may affect that. But it definitely opens the door for questioning and searching for a greater answer.
I actually have two questions. One is to you, I don't know your name.
Cori.
Cori. So how can you tell the calcium released from bacteria, not the host cell, like human cells? Because we probably do not want to calcium released by our own cells, but only released from only bacteria, so we can actually observe the calcium from the bacteria, so we can use it. So how can you tell those difference between the calcium released from bacteria and from host? And there's one question, and I'll have
So the question is, how can we tell the difference between whether or not the calcium comes from the microbe or from ourselves, from our diet?
right.
So that's actually a really interesting question. So in germ-free animal studies, a germ-free mouse will absorb considerably more calcium than an animal that has microbes living in its gut. So the microbes do sequester some of these minerals for their own benefit, but even in a model where we feed prebiotics, obviously the microbes would take some of that, but we're still able to see that calcium going to the human and going specifically to the skeleton, which is where we want to see it. And we can measure that with stable calcium isotopes.
From actual bone, host bone? Or,
Mhm.
Oh, okay, I see. Yeah, thank you.
You're welcome.
Can I question over here?
Yeah, so I was interested in how you target the neoantigens and RNA. So correct me if I'm wrong, but that's inside the cell, right? It's not expressed on the outside of the cell. So how did you go about targeting those if they were inside the cell? And from there, how did you kill the cancer once it's targeted?
So the question is about the mechanism for these personalized cancer vaccines. Whether the mutation is encoded in the DNA or it's only made in mistakes in making the RNA, the peptides that result from that are presented on the outside of the tumor cell. And so those are the recognition signals for the T cells, the host T cells, to come in and kill that tumor cell. Is that okay?
Yeah, so the T cells were able to recognize it, so you had to make them create that antigen.
Yeah, you treat it just like a vaccine for infectious disease. So you take those, so the question is, how did you get the T cells to do that? You take the peptides that would be encoded by these mistakes in the tumor cell, and you actually vaccinate the animal, or eventually the person, in order to raise the T cells to go after the tumor cell.
And how'd you deal with, like, so there's usually some tumor inhibition, right? How did you get around that?
That's a good question. So the question is, how do you get around tumor inhibition? In the clinical trials that were just concluded, they gave the vaccine directly. So just the vaccine to the individuals was okay, although these people had been debulked of their tumors. People have also done it where they've vaccinated with these checkpoint inhibitors. And those checkpoint inhibitors basically take the brakes off your immune system and let it rev up.
I want to ask a more general question to all of you. I'll start with Beata. When it comes to a personalized or precision approach to the disease that you study with the speech and hearing deficits, what do you see to be capable or possible in 5 years? And then what do you think will be possible in 15 years in your field?
I think I'd like to start with the audiologists. I think they're the ones that have the greatest ability to use what they know already and implement it in clinical practice. And their greatest need is going to be education. So we polled 4,000 speech language pathologists and audiologists through the American Society of American Speech Language Hearing. I have so many acronyms floating through my head. But the ASHA, the American Speech Language Hearing Association, we polled 4,000 audiologists and speech language pathologists. And they all have a feeling that, yes, genetics will be extremely important in the future, but they also rated their own competency very, very low. So in 5 years, I hope to get our clinical professionals to a point where they have a real workable, solid background in genetics so that they can meet somebody and figure whether there's a genetic pathology going on and translate that into, especially in audiology, where, you know, knowing somebody's genotype for hearing impairment will be important for whether the person will be a candidate for cochlear implants or not. You can use that in very practical ways. So that's one thing. Another thing that I would love to see is development of preventative courses of treatment. Currently, under insurance laws, we can only treat children, individuals, once they show a deficit or a delay in an area. But once we have a pretty good solid idea that this child is at a high genetic risk, for instance, in galactosemia or maybe even other non-syndromic kinds of communication disorders, you know, bringing online a set of preventative courses of action like our Babel boot camp, if we can show that it works better than the wait and see and then treat approach, that's where I want to drive the boat.
Oh, so in 5 years, personalized nutrition. I don't think we'll be there. I think there are some things that are there. So if someone has PKU or phenylketonuria, that's a nutritional disease where someone can't digest phenylalanine, the amino acid. So we have nutritional approaches to treat that, and babies are tested shortly after birth. So that's a good example of a place that we're already there. Another good example would be folate metabolism. So people have been tearing apart the folate cycle for decades now, and we really know which proteins tend to have issues or enzymes that don't convert properly, and so we know who may need a higher dose of folate to optimize those pathways. But in terms of, and again, this is treating diseases and illnesses, but what about, we all eat to live, we love to eat, and how do we optimize our nutrition just for general well-being and to prevent having cardiometabolic diseases later in life? I think that's a huge gap, and the Academy of Nutrition and Dietetics, their consensus statement on whether or not precision or personalized nutrition is foreseeable anytime soon is thinking more like 20 to 25 years out. We'll have some better data for that, but it's a work in progress.
Wow, that's great.
Okay, that's an interesting question. I think the most exciting thing that'll probably be out there within 5 years is that we'll see the first example of using CRISPR to do a gene therapy. And it's probably going to be in China first. And it's not going to be the most, people argue against gene therapy partly, and I used to, because each single gene defect, disease, only accounts for a very small amount of the number of people. However, if you have a general systematic way to do that gene therapy for each one, no matter what the gene is, it could actually have an important impact, because if you add up all the 900 and so single gene effects of people, you add them all up, it's actually a substantial number of the medical community treatments. So that will be very exciting, but it won't be the most impactful. I think the most impactful thing would be for people developing the biomarkers to do early detection of disease, because it's already there with standard therapy for almost every disease except Alzheimer's. The existing therapies can have a huge impact if the disease is discovered early, both on the livelihood, the life expectancy, as well as the economic costs. So I think that'll be the most exciting thing in 5 years. So
I'm going to get you to write letters of support for my grant applications because that's the kind of work that I do. Another question. Yeah, Ji-san.
Yeah, I have a question about the in vivo and in vitro study model. So, in order to develop whatever the method of the remedy, we have to do some kind of research and then consider the nutrition, behavior, and even personalized the medicine. Um, even without considering that personalized medicine, we have a lot of challenges that having a difficulty with a difficulty to have the good research model, in vitro model or in vivo model. So I'd like to hear about your opinion about that because all we scientists know those challenges that a lot of we use cell culture, we use animal, but we know that there's difference. The animal anatomy and the human anatomy is totally different, and then also the cell culture. A lot of cases, there's of course a lot of cases, whatever experimental result in the lab doesn't actually mimic in reality. So now we are now going to personalize the directions. Now we have even more challenges. So what do you use as whatever host or whatever research model? And then that is one. And the second, what?
Oh, we'll address that question first. Okay. So the question was, in a nutshell, one of the big challenges in developing new therapies, period, is having good model systems to develop these therapies in, whether it's tissue culture or animal models. And when it comes to now taking it into a personalized or precision medicine approach, if we already have insufficient models for generalized medical approaches, how can we deal with that with precision medicine?
I'll just make a really brief comment. So in my field, speech and reading and language, you might imagine that an animal model is a little bit challenging, but believe it or not, the FOXP2 gene, which is the first gene that was validated to cause a severe speech and language difficulty in humans, has been actually studied in mice and in zebra finches. So where they can't learn the song if the gene has been incapacitated. So there's huge low-hanging fruits to be picked still. That work is still in the very beginnings, but I'll let you speak a bit more.
So two things. One, I think that, and we've already seen some of this, the best thing to do is to get into humans first. To just don't think around with mice. Get into humans as fast as you can. And I think we're increasingly seeing that tendency, the recognition that you want to get into humans. And they're developing new ways to enable that. For us personally, we try not to use models anymore. We use dogs because dogs are not models. Dogs, anything you can make money off of is not a model. So our cancer vaccines, we're doing this in a dog trial. The trial for the protective cancer vaccine is going to be in 1,000 dogs, owner-enrolled dogs. And I think not for all diseases, but for many diseases, dogs are a very good system to work in, and if you're successful, you make money.
Oh gosh, I don't know if I have an answer to this question. I mean, in our area of work, we use human models and animal models, and it's helpful also that if you can do something in a human model, you can't always answer the mechanistic questions, but if you can go back to those earlier cell culture or animal models, it is so informative on speculating about mechanisms, and then you can devise plans to actually then test that in a human model. And so I think for me, we need all of it, and it just is a feeding loop that keeps coming back to itself.
Other questions?
Can I add it to
Okay, sure, Ji-san.
Yeah. Yeah. So yeah, the reason that I am asking, I asked those questions is because I can actually see the challenges because I am doing like a 3D cell culture and also to use as a model for the infectious disease study. So I can actually see what is really challenging in real clinical, like in real world. So I understand. Yeah. So doctor actually mentioned the dog like it's not model, but still it is not human. And even we already talked about individual has a difficult individuals are different. So when we have a personalized medicine, that one does not work to other person anymore. So that's why we are more now people are more interested in more personalized medicine. So which means that still the whatever if it is model or not, still we have some kind of challenges. So now the question is, what do you see? What the most challenging part for in this field? What do you think that we need to have more in order to advance this field, like tools or whatever ethics or any kind of thing that you can actually think of? What do we need to advance this field?
Okay, yeah, so in a nutshell, Jisan is asking what are the barriers right now to moving a personalized or precision medicine approach forward, and what tools or what resources or what ethical considerations should we be developing or using to bring down those barriers?
I don't know that I can speak to tools, but in terms of barriers, our current healthcare system is very public health and population focused. So when we know that something works, while it may not work for everyone, we can mobilize resources to treat everyone with a vaccine, or offer resources to everyone that may or may not be helpful with the hope that they will be. And so I think from a policy standpoint, we need to get policymakers on the same page as those working in the precision medicine arena, so that we can begin to hone current policies so that that paradigm fits in how we care for the population.
Yeah, I think for us it would be clinical trials. That would be a lot of work, a lot of funding necessary to develop interventions that are targeted to an underlying endophenotype, and also earliest interventions. Those things have to be test driven first, and that's going to be a huge challenge.
So the standard answer is money, right? So, but I'm going to say that I don't even think that that's actually the biggest limitation. I think it's guts, because I think the research community is a little spineless in terms of trying to take chances and do important things that count. And if you look at it, all of the drug trials that we've done over the last 10 years, you can barely tell the effect of the drugs from the controls. So that says that we're not taking many chances. We're not doing the kinds of experiments that we really should be doing in terms of breaking things through. We're doing mundane stuff.
I would actually comment, though, that it's also possible that we are trying things, but we fundamentally don't understand the problems we are trying to solve. And that is what leads to this discrepancy of like, oh, we try all these things and the placebo versus the actual drug is such a small difference, because we don't have a right grasp of the problems we are trying to solve.
Yeah, the comment from the floor was about lack of fundamental understanding of some of these diseases that is underlying some of this. Joe.
So part of understanding those fundamental processes is using models. And I think George Bach said all models are wrong, but some are useful, right? And so that's where we are. The entire field of immune checkpoint blockade started with Dan Barber infecting a PD-1 deficient mouse, and it died. And that's where that entire field came from. So I don't think that we should ignore the models, but certainly they can be useful. My question is mainly directed at Stefan. As you develop a cancer and you start increasing the number of neoantigens, we now know you're also increasing, or you can increase also the aggressiveness of the tumor. Do you see any segregation of neoantigens in particular tumors with the aggressiveness, and do you ever run into the case where you target the wrong cells in a heterogeneous tumor?
So the question is, do we pick the wrong neoantigens?
Well, by targeting neoantigens, you eliminate bulk of the tumor and then give rise to more resistant or more aggressive forms of the cancer.
Okay, so do you get more aggressive tumors in some cases? We have not seen that yet. In the human checkpoint inhibitor trials, it's starting to come back that some of the people that are getting a reoccurrence, they seem to be getting a more aggressive tumor coming back the second time. And that may be this case, is that you've forced the immune system to create a certain area of protection, and any other tumor is going to have to come outside of that umbrella in order to come back. And I think the data is early on that yet. Is that okay? Is that what you're asking?
Go ahead, Fred.
Can I go back to the idea of models? One of the things that we do in my lab is we love inbred mouse models because we don't have to worry about all the heterogeneity that you guys have to worry about. But that then takes away from being able to look at genetic differences unless we put the genetic difference into the animal, like knocking out a gene. We only know to do that because we think the gene is important. So how do we, it's sort of a lot of circular reasoning. Are there better models that we can come up with that allow us to have all the animals acting the same, but still be able to look at genetics? And so that's a question I have. I think there are a couple models. Dog might be one of them because you have a really good understanding of the genetics of different breeds of dogs.
Is that a question?
That's a question. What models could we, what better models could we have that both allow us uniformity, but then allow us to look at genetic components easily?
I don't know. And what Joe just said is true. I'm not arguing that, this isn't all or nothing kind of thing. And I think each case that you're studying, you're going to have to look carefully to see what it is. For cancer, there's no doubt in my mind it's better to get into dogs right away. You'll know whether it'll work or not in people. But for other things, I'm not sure what the best model is. I think in general, though, I think we haven't thought creatively enough about how to get it into people early, as soon as we can, and then see what's interesting, and then go back and try and figure out the mechanisms. As you know, we don't know how most vaccines worked. They worked. And they saved hundreds of millions of lives, and we still don't know how some of them work.
I'm going to wrap up with one last question for our panel, and I appreciate Stefan talking about this in the prologue, in that there are some severe ethical considerations when it comes to taking these technologies that we're developing out to the rest of the world, not just to rich nations. So what specific concerns in each of your fields do you have about taking this out to other underserved populations, or even underserved populations within our own country? Because certainly most of medicine was developed for Caucasians and males and younger people.
Well, I think for my field, that would mean people who are going to benefit will have to give us their DNA. And so confidentiality, GINA, so the Genetics Information Non-Discrimination Act is constantly being discussed. How do we balance the need to have people's genomes against, some of the negative effects that it could have in their lives? Could they be denied long-term care insurance or life insurance? So these kinds of, that's what I see in directly dealing with the people that come to me for research purposes is, the worry, what will you do with my DNA, and can I have some kind of negative consequence from that? So that's a worry that I have.
In terms of culture and different sorts of communities and populations that may be more disadvantaged than a U.S. population, I think it's always important to consider why a community does the things that they do. So nutritional practices and dietary practices are heavily embedded in our culture. And so if we know that a certain food or nutraceutical could have a meaningful impact, but it is derived from a food component that culture would not eat, it's probably not ethical for us to go in and try to push it upon them and to find alternative solutions. and that can get, hairy. But it's an interesting thing, and it's kind of fun to be able to work around those sorts of problems because it's not just biomedical, it's also cultural and environmental. And I think that's really why I like working with humans, because they're just so different and unique, and you get to do that.
So I always am amused by this argument about who's going to get my DNA. This is the same people that will take their credit card and use it on a daily basis and give somebody the access to all of their finances. It's just amazing. But I think one of the things that I don't know this area, but I think that for a lot of these things that are going to be developed that may be relatively expensive, I think but they're on a cost-benefit analysis. If you do the cost-benefit analysis, you can see how good it could do. I think people are going to have to come up with creative win-win situations. The idea of just giving somebody something usually historically has not worked that well. But if you can figure out win-win situations where people can have these advantages, but it's not charity, I think, and there are models for that. I think that will be a big important thing for getting more distribution of these technologies.
All of our panelists and questions from our audience today. Thank you so much.